A very good afternoon everyone. I am Dr. Swati Sharma from DRC, University of Delhi. Today we would be studying Foundation of Microbiology. So this is the part 2 of this syllabus. Last time we covered the Foundation of Microbiology, in which we studied the different contributions by different scientists and how they laid the foundation of the microbiology. But today we are going to explore the fascinating world of the organisms, which are so tiny and small that we cannot see them with our naked eyes, and we need to use the microscope to visualize them, and that is why they are called microbes and the field of study is called microbiology. But they are so small and yet they control many aspects of our life. The subject of today is called the Foundation of the Microbiology, and by the end of this lecture you should be able to know what are microbes, and you will able to realize that microbes are not just agent of the disease, they are essential for the life on the earth. By the end of this lecture you should also able to know what is microbe, to understand the basics of microbiology and understand and identify major groups of microbes, learn how microbiology was developed as a field of the science, and recognize why microbes are important to humans and the environment. So now you know the field of study of microbes is called microbiology, and it requires use of microscope. The main microorganism can be categorized into 5 different groups, which is bacteria, archaea, fungi, algae and protozoa. Most of them are disease causing, but most of them are also the habitat of our natural body system. For example, our gut has microbes. The bacteria which is able to synthesize vitamin B12 complex, and hence it is in the symbiotic relation with ourselves. The bread we consume, the curd we eat, all of them have rich microbial community, and that's why it's important to study them, and they are important not only to us but to the environment as well. So as you can see in this picture demonstrates the scale of an eye. See, a human eye has a limitation. We cannot see beyond 100 micrometer. So 100 micrometer is the range of the human eye. Anything below 100 micrometer cannot be seen with the naked eye, and we need to use the microscope to see it. So if you can see it in the slide that all the microorganisms, they can be visualized under the microscope, but certain microorganisms such as the fungi, pores or algae, they can be seen with the naked eyes as well, but still they are considered microorganisms because their spores are so tiny and they cannot be seen with the naked eyes. So this picture depicts the range of the human eye and the different size range of the different microbes, including your fungi, algae, bacteria and protozoa and viruses. So microbiology is the scientific study of the microorganism. These microorganisms are also termed as microbes, and these organisms are too small to be seen without a microscope. But do not mistake their size for the lack of importance. They are everywhere. Every place you can think of is infested with microbes. The curd you eat, it has lactobacillus. The bread you eat, it has a fungi called yeast. The medicines you take, for example the antibiotics, they all are derived from the fungi called penicillium. Even the oxygen that we breathe, it is enriched in the microorganisms. So all are connected to the microbes and all are rich in the microbes, and thus microbes are there in the environment. They can thrive in any environment depending upon the pH and the temperature, and they are crucial for maintaining the environment on the earth. But several microbes are disease causing also, such as SARS-CoV, such as HIV, or protozoas, such as the protozoa causing malaria. So it's very important to study them and demarcate them. So in order to study them, we first need to understand the growth conditions of them. So this table illustrates the different growth parameters and how they proceed. So now let us first understand the growth of the microbes in different culture. First of all, you need to know why we need to culture the microbes. In order to understand how they are growing and how we can use them, we need to study the growth curve of the microbes. Microbes are usually grown in the culture. This culture can be either liquid culture or the solid culture. So in liquid culture, the growth is exponential and you cannot find a pure culture. But when you have to isolate a pure culture from the microorganism, you usually culture it on the agar plate or the solid culture. Then the single colonies can be picked. So in order to grow different microbes in the culture, we need to first understand their growth curve. So this is the typical growth curve of microbes under the suitable condition. There are 4 different distinct phases. With the passage of time, each phase comes. So the first phase is called the lag phase. As you can see it in the graph, the lag phase is steady. You do not see any growth in that phase, but it does not mean that microbes are inactive during that phase. इस phase में भी बहुत ज़्यादा microbial activity होती है. इस phase में microbes prepare करते हैं अपने आप को ताकि वो अपने colonies को double कर सकें. And most of the microbes, they divide asexually by making multiple copies of themselves. So during lag phase, they replicate their genome and they get double the size. And in exponential phase, you see a exponential growth in the number of the microbes. So this exponential growth is because of the division of the microbes. And now once they have synthesized the genetic material and doubled the genome during the lag phase, they can divide and multiply themselves during the exponential growth phase. So this is the growth phase of the microbes. But once they reach the exponential growth phase, they start to utilize the nutrients present in the culture media. So all the microbes, they are grown in a certain culture media, and that culture media contain nutrients. So when the cells are dividing, these nutrients are being used by the cells. So soon these nutrients start depleting, and then the rate of growth becomes equal to the rate of the death of the cells. So that remarks your stationary phase. So during stationary phase, the number of the cells produced is equal to the number of the cells which are dying, and then the growth rate is a plateau. But once all the nutrients are being used, there is a sharp decline in the number of the cells. So the cells start to decrease, and this is marked by the death phase. In death phase, there is no growth of the microbes and the number of the microbes start decreasing. So it's very important to understand this growth curve, because in industry we need to culture the microbes. As I told you, these microbes are very much important in the industry. We use them in curd and cheese industry. A bacteria called lactobacillus is used in the curd industry for the coagulation of the milk after the pasteurization of the milk. The fungus yeast is very important in brewing industry. Brewing industry means conversion of maltose or malose or the fruit sugars into alcohol. Then yeast is also used in bakery. So it's very important to understand these growth curves and the duration of the each phase of these growth curves so that you can exploit the growth. We want to accumulate a product and minimize the accumulation of the toxin products during the growth curve. So it's of huge importance that we understand the growth curve for each microorganism that we want to use in our industry, and the choice also depends upon which industry we are working on. So growth curve is actually the population growth study, and it is studied by analyzing the growth curve of a microbial culture. When the microorganisms are cultivated in liquid medium, they are usually grown in the batch culture or a closed system. That is, they are incubated in a closed culture vessel with a single batch of a medium. So see, I told you that cultures can be of 2 different types. First is your solid culture, in which you use the agar plates, and in agar plates you can have the single colonies for the pure culture, but agar plates cannot be used for the upscaling. For that you need to use the liquid culture or the liquid medium. So in liquid medium, again the growth can be of 2 types. First is your batch culture, which is also termed as closed system. In batch culture, you take the media rich in the nutrients and then you inoculate it with the particular colony. and then you close the system. That is why it is called the closed system. So you do not open the system in between. You open the system at the end when the products is accumulated and all the nutrients are being depleted. The other type of the culture which uses liquid media can be your continuous culture. As the name suggests, a continuous culture is an open system because you will be continually adding something and continuously removing something. So in continuous culture, you take the nutrient media and you keep on adding the nutrients after a certain period of time, and you keep on taking out the product or the byproduct at particular period of time. So in your continuous culture or your open system culture, you receive a constant supply of your product. So the yield is always high in the continuous system, but as the continuous system is the open system, the chances of infection or the chances of contaminations are more in the open system or the continuous culture system. So that is the limitation of the continuous system. So in industry, specifically in the bakery industry or in the brewery industry, people prefer continuous or open system because they want more yield. But in the pharma industry, where you do not want any batch to batch variation and you want your all the pharmaceutical products such as antibiotic, such as your medicine, such as your insulin to be same, you use closed culture or batch culture as it minimizes the infection or the contamination. Now let's move further. The growth of the microorganism reproducing by the binary fission can be plotted as the logarithm of the number of the viable cell versus the incubation time. The resulting curve has 4 distinct phases. So this is the technical definition of the growth curve. You define the growth curve as the growth of the microorganism which reproduce asexually by binary fission, because most of the microorganisms, they are prokaryotes. Prokaryote means they have simple biological structure. They do not have any well-defined nucleus, and all their cellular organelles are present in the cytoplasm without any membrane covering. So the best way for them to reproduce is by just dividing themselves, and this division is called as binary fission, in which first they make a copy of their genome, they ideally double their genome, and then they divide the genome in 2 equal parts and thus form 2 copies of one cell. So if you plot the graph of the number of the viable cells, viable means living cells, the number of the viable cells versus the incubation time as the log scale, then you will get the growth curve. And as I told you, the resulting growth curve have 4 distinct phases. Let us revise what are these 4 distinct phases. First is your lag phase, then is your log phase, then you have your stationary phase, then you have your death phase or your decline phase. So now let us first understand what is the lag phase. When the microorganisms are introduced into the fresh culture media, with usually no intermediate increase in the cell number occurs, and therefore this period is called as the lag phase. क्योंकि अभी आपने एक बस fresh culture medium लिया है, उस culture medium में आपने you have provided all the nutrient source such as the source for the carbon, nitrogen, phosphorus and all the essential minerals including your magnesium, calcium, iron, along with aeration. Aeration means presence of oxygen, because most of these organisms, they need oxygen to divide and grow. So when you introduce a new microorganism or a new inoculum in a fresh culture media, you do not see any immediate increase in the cell number, and that is why this period is called lag. It means the growth is lagging behind, but the name shows that there is no growth taking place, but this is not the case. Although the cell division is not taking place right away, but there is net increase in the cell component. The cell is synthesizing new component, and that component is basically the genome. So it can either be the RNA or it can be the DNA, depending upon which microbe you are culturing. A lag phase prior to the start of the cell division can be necessary for the variety of reasons. So some people may think that lag phase is the wastage of time, we do not need that phase and we can accelerate the cell division by shortening the lag phase. But it's not true. A lag phase is very crucial and it's very important for the cell division, because the cell is under the S cycle, which is the synthesis cycle, and it is continuously synthesizing all the proteins which are needed for the cell division. So it is therefore important for, because of the variety of the reasons. So you can see these two graphs. These graphs depict the growth curve of the continuous nature. So they are the lag phase. So you see the difference in graph which is at the left and which is at the right. So these graphs show the difference between how the lag phase or the growth phase can look like. So now let's move to the exponential phase. Once the cells has synthesized the sufficient amount of DNA and the sufficient amount of the proteins which are needed by the cell to divide, the cell enters the exponential phase. During exponential phase, the microorganisms grow and divide at the maximum rate possible to given their genetic potential and the nature of the medium and the condition under which they are growing. So exponential phase is also termed as the log phase. In this phase, the cell divide rapidly according to their genetic potential. For example, for E. coli, the division time is 20 minutes. So if you are inoculating E. coli in the culture media, the cell will divide after every 20 minutes, and after every 20 minutes you will have double the number of the cell. So if you plot the graph of the time versus the number of the cells, it would be on the log scale, because after every 20 minutes there is doubling. And it also depends upon the nature of the medium. Your medium should have sufficient nutrients. If your medium or the culture in which you are growing your microorganism lacks certain nutrients, the cell will not grow, and the conditions under which they are growing. So when I say condition, there are different conditions such as condition of pH, condition of temperature, pressure, presence of oxygen or absence of oxygen, because all these microorganisms are different. They differ in their need of optimum pressure, temperature, pH and oxygen concentration. So depending upon what microorganism you are using as an inoculum, you decide the conditions of the growth. And the growth rate is constant during the exponential phase. So first they start dividing rapidly, but then the growth rate get constant depending upon the generational time. Generational time is defined as the time taken by a single microbe to double its population, and this generation time differ for each microorganism. So that is that microorganisms are dividing and doubling in number at a regular interval, because each individual divides at a slightly different moment and different rate. The growth curve rises smoothly after then rather than in discrete jumps. So you do not see the peaks or jumps like that. You just see a single line exponentially rising because they have a definitive generational time. So the population is most uniform in the terms of the chemical and physiological properties during this phase. Therefore, exponential phases are usually used in biochemical and physiological studies. Exponential growth phase is the balanced growth phase. That is, all cell constituents are manufactured at a constant rate relative to each other. If nutrient levels or other environmental conditions change, unbalanced growth result. So see, now you have to apply your understanding of your continuous culture and batch culture here. So in batch culture, the growth rate is balanced because the batch culture is a closed system. So you have provided everything at once and the growth is being balanced. Nothing is changing except from the nutrients, because nutrients are being utilized. But in continuous culture, you are providing everything at a constant rate. You are replenishing the nutrients. You are providing the nutrients what all are limiting at a constant interval of time. So the cells are able to utilize it, and then you see the rate change in the growth. So this is the growth during which the rate of synthesis of the cell components are very relative to one another until a new balanced rate is reached. This response is readily observed in a shift-up experiment in which bacteria are transferred from a nutritionally poor medium to a richer one. The cells first construct new ribosome to enhance their capacity for protein synthesis. This is followed by increase in protein and DNA synthesis. So, growth rate is still maintained constant, but it does not mean that there is no shifting in the balance. So, once the nutrients start to get exploited, now either you replace the medium with the new nutrient, you provide the richer one, or you let the cell grow in that media. So what will happen? The cell first will make new ribosome because now they need more protein to survive, and then they will make more protein and more DNA synthesis. And as the more DNA synthesis will happen, what will happen? The cells are again to divide. So now they will start to divide again. So as you can see, this is the different conditions on which the growth occur. You can see a linear straight line which is expected outcome, and you can see the curved line also, and above you can see the different generation time for the different microbes. So now after the exponential phase there is stationary phase. So once the growth has reached the maximum potential, every population growth will cease. Cease mean it will stop. The growth curve will become horizontal. So once the growth curve become horizontal, this is termed as stationary phase. So this stationary phase is usually attained by bacteria at a population level of around 10 raised to power 9 cells per ml. Other microorganisms do not usually reach this high population densities. Protozoans and algal culture often have maximum concentration of about 10 million cells per ml, because algal cultures are very difficult to manage, so they do not reach that much concentration. But bacterial culture, they are easy to culture, and bacteria are the simplest prokaryotes. So you can see the high level, that is 10 raised to power 9 cells per milliliter. And during the stationary phase, the growth curves became stagnant, and the last or the fourth phase is the death phase. In death phase, there is detrimental environmental changes like nutrient deprivation and the buildup of toxic waste, which lead to decline in the number of the viable cell, which is the characteristic of the death phase. So we will continue after this. Thank you.